From the smallest bacterium to the largest whale, all living things are made of cells. These tiny building blocks are constantly growing, dividing and dying. This ongoing cycle of cell division is essential for life itself. It allows organisms to grow, repair damage and reproduce. Imagine a bustling city where old buildings are torn down and new ones are built in their place. That's what's happening inside you right now on a microscopic scale. Without cell division, wounds wouldn't heal, bones wouldn't mend, and you wouldn't have grown from a single fertilized egg into the complex being you are today. There are two main types of cell division, mitosis and meiosis. Both involve the duplication and distribution of genetic material, but they have different roles and outcomes. Understanding these processes is like unlocking a fundamental secret of life. It allows us to appreciate the intricate dance of cells that keeps us alive and allows for the continuation of our species. It's a story written in the language of DNA, a story that unfolds with every cell division. Think of a busy office where important documents need to be copied and distributed to everyone. That's essentially what mitosis is all about, making sure each new cell gets a complete set of instructions. This process is used for growth, repair and asexual reproduction in many organisms. Mitosis is a continuous process, but to understand it better, scientists have divided it into distinct phases: Prophase, Metaphase, Anaphase and Telophase. Let's break down each stage. Imagine you're moving. Before you can pack boxes, you need to gather everything together. In Prophase, the cell does something similar. The DNA inside the nucleus, normally a jumbled mess of genetic material, condenses into organized structures called chromosomes. These X-shaped structures are easier to move and divide than loose DNA strands. Now it's time to line up those boxes for loading. In metaphase, the chromosomes line up neatly at the cell's equator, like soldiers reporting for duty. This ensures that when the cell divides, each new cell receives an equal share of the genetic material. Anaphase, splitting up the goods. Once the chromosomes are lined up, it's time to divide. During anaphase, the chromosomes split at their center and the two halves, called sister chromatids, are pulled apart to opposite ends of the cell. This ensures that each new cell gets a complete set of chromosomes. Telophase, two new homes. With the chromosomes separated, it's time to pack them into their new homes. In telophase, a new nuclear envelope forms around each set of chromosomes, creating two new nuclei. The cell then divides its cytoplasm, the jelly-like substance that fills the cell, in a process called cytokinesis. The result? Two identical daughter cells, each with a full set of chromosomes. Meiosis, shuffling the deck for the next generation. While mitosis is about creating identical copies, meiosis is all about diversity. This type of cell division is responsible for sexual reproduction and generates sperm and egg cells, also known as gametes. Meiosis involves 2 rounds of cell division, resulting in 4 daughter cells, each with half the number of chromosomes as the parent cell. These cells are not identical to each other or the parent cell, contributing to genetic variation. Meiosis 1, halving the chromosome set. The first round of meiosis is where the magic of genetic shuffling really happens. Remember how chromosomes come in pairs, one from each parent? During Prophase Y, these homologous chromosomes pair up and exchange segments of DNA in a process called crossing over. It's like shuffling 2 decks of cards, ensuring that the offspring inherit a unique combination of genes. Meiosis I then proceeds through Metaphase 1, Anaphase Y and Telophase Y, much like mitosis, but with a key difference. Instead of sister chromatids separating, entire homologous chromosomes are pulled apart, resulting in 2 daughter cells with half the number of chromosomes as the parent cell. Meiosis 2, separating sister chromatids. The second round of meiosis is more straightforward, resembling mitosis in many ways. The two daughter cells from meiosis I undergo another round of division, separating the sister chromatids. The result, 4 genetically unique haploid cells, each carrying half the number of chromosomes as the original parent cell. Mitosis versus meiosis. A tale of two divisions. Mitosis and meiosis are both essential for life, but they have different purposes and outcomes. Mitosis is like making photocopies. It produces two identical daughter cells from a single parent cell. Meiosis, on the other hand, is like shuffling a deck of cards. It generates 4 genetically diverse daughter cells, each with a unique combination of genes. Think of mitosis as the workhorse of the body, responsible for growth, repair, and asexual reproduction. Meiosis, on the other hand, is the master of diversity, ensuring that offspring inherit a unique combination of genes from their parents. This genetic variation is essential for adaptation and evolution. While mitosis occurs in most body cells, meiosis is restricted to specialized cells in the ovaries and testes that produce gametes. Understanding the differences between these two processes is crucial for understanding the fundamentals of genetics, heredity, and the diversity of life on Earth. Body cells versus germ cells, specialists in different fields. Our bodies are made up of trillions of cells, but not all cells are created equal. We can broadly categorize them into 2 main types: somatic cells, also known as body cells, and germ cells, which are the precursors to sperm and egg cells. Body cells are the jack-of-all-trades of the cellular world, responsible for a wide range of functions, from contracting muscles to transmitting nerve impulses to secreting hormones. They undergo mitosis to replenish and repair tissues throughout our lives. Germ cells, on the other hand, have a much more specialized role to transmit genetic information to the next generation. They are found in the ovaries and testes and undergo meiosis to produce gametes. Imagine a large corporation. Body cells are like the many departments: sales, marketing, finance, each with its specific role in keeping the company running. Germ cells are like the research and development department, focused on innovation and the future of the company. Diploid and haploid, the chromosome count. Chromosomes are like instruction manuals for building and maintaining an organism. They come in pairs, with 1 chromosome from each parent. The total number of chromosomes in a cell is called the ploidy. Body cells are diploid, meaning they contain 2 sets of chromosomes. In humans, this means each body cell has 46 chromosomes, 23 pairs. This diploid state is essential for normal development and function. Germ cells, on the other hand, are haploid, meaning they contain only 1 set of chromosomes. In humans, this means each sperm and egg cell has 23 chromosomes. Why is this difference important? When a sperm fertilizes an egg, the 2 haploid cells fuse, restoring the diploid number of chromosomes in the zygote, the first cell of the new individual. This ensures that the offspring inherits a complete set of genetic information from both parents. Conclusion. The dance of life continues. From the single-celled organisms that first appeared on Earth billions of years ago to the complex multicellular life forms that inhabit our planet today, cell division is the fundamental process that drives growth, development, and reproduction. Mitosis ensures that our bodies can grow and repair themselves, while meiosis shuffles the genetic deck, generating the diversity that makes evolution possible. Understanding the intricacies of mitosis and meiosis allows us to unravel the mysteries of inheritance, appreciate the delicate balance of life, and marvel at the elegance of the cellular world. It's a reminder that even at the smallest level, life is a symphony of coordinated events, a dance of molecules and cells that has been going on for billions of years.